who join us for the Agile Leaders events. And our goal, this is the first one of the year. Last year we did 100, and I think we're in the end of our whole events, we did about 120. So it was a marathon year, and we're going to continue exciting you and bringing you great ways of spending a few hours doing learning stuff, engaging. These are cameras on, so you never know when we might pick on you to make a comment, because it's all about relationships, and entertaining and making sure that everybody's connected. So this is what things, keeping people connected. And in the Agile Lead events is our, if you like, our attempt to bring in thought leaders, people who know what's going on, and people who have got experience what's happening, you know, in various parts of the world, and bringing them to us so that we can be inspired, we can model our businesses, and we can make contacts for business development. So the Agile Leaders is our great event, and this is part of our, we've got two parts to this one, and it's a memorial, if you like, event for JP Morgan, which all of us would normally be at. And last year I had a terrible experience of leaving all my shirts in the wardrobe. Unfortunately, I decided on day one I would put on a formal shirt, and I left them all there up this year, but I think they're now being sold. So that's a very sad thing. But JP Morgan is a great time when we meet everybody. It's the beginning of the year. It's a sort of it's a milestone to what's going to happen. And that's really what we want, some of the essence of our events this week. And part one is going to be mainly on cell and gene therapy and BD. We've got two fantastic groups of people to chew over various areas. And then Friday, we're going to have the legendary Max Herman, and I think we're getting Sue Sue Charles, isn't she, Graham? And we're gonna do a bit of a roundup of what's happened in JP Morgan, and what are the highlights, what's the soupçon of going on. Yeah. So without further ado, Graham,
I would just like to say, yeah, no, I mean, you know, obviously we've been able to do this very cheaply, you know, get you all together, and that's through the kind sponsorship of Cobra Biologics. And you know, it's great working with them. You're going to be meeting them here today, as well as our other fantastic guests. So if you'd like to continue, Tony.
Yeah, so we've got the way we're going to structure the day, the time we're going to have one session on cell and gene therapy and what's hot. We're going to have a session on BD and what's the feeling of being a BD person in the current world. And then we're going to do a bit of serendipity networking, Graham. And what's the time frame for the whole event?
Yeah, so we're going to have this discussion. It's going to go for about 45 minutes with our panel, and we're going to basically work it. We'll have some questions and answers at the end. So if you've got any questions during the panel, if you put them into the chat, and then, you know, we'll curate those and get you to ask those questions towards the end. In the last kind of 15 minutes of the first hour. Then we're going to have a 10-minute break. And then we'll have a small panel just talking about doing BD, the challenges of doing BD virtually. And then we're going to have 30 minutes of breakout rooms right at the end, so you get a chance to get up closer and personal with everybody.
Yeah, and without further ado, let's move on to our first group. And I've got a fantastic group of people who, and basically the idea, what's happened in 2020? Well, what's happened in 2020 is that cell and gene therapy probably remains the top of the list of the life sciences exciting areas. And there's a lot of development about what is cell and gene therapy, and a lot of morphing into various aspects of what it is. And you know, then there's been the tremendous work, of course, on COVID, and then there's this sort of idea of what is a vaccine, and I think that there's a lot of interest, actually, what is a vaccine now? You know, is it a bit of a prophylactic therapeutic? What is it? So I think there's a lot of discussion, and I think our group of people who I'd like to introduce will go round, and I'll introduce you to. And Graham, if I miss anyone, please help me. But I'd like to introduce you to our great group of people. And first, we've got Arvind, and maybe Arvind, you could just say who you are and what you do briefly.
So I'm Arvind Hundal, an ex-molecular biologist, a gene jock originally. Been in business development for a really long time. I don't even say seasoned because it just says I'm old. And currently I'm at BioNtech, obviously working on the coronavirus vaccine. And it's been my obsession for the last more than 12 months, actually. I think we started before the beginning of 2020, we started the R work. So it's been a long haul.
Fantastic. Welcome, Arvind. And
Thank you.
I've had the vaccine, so I've got a bit of your stuff in my left shoulder.
Oh, it's so exciting to actually see somebody who got it. Yay.
And he hasn't got a third head.
There was a bit left over because they couldn't freeze it. So the doctor said, well, you better use it, Tony. So it was a case of who you knew actually, and that's how I got it.
So now we've upped it to 6 doses per vial, so you've got probably a full dose, which is good.
Ah, very good. Well, then moving on to the next one, which is Philip. Welcome, Philip.
Hi, Tony. Thank you. Thank you. Yes, I'm Philip Ridley-Smith. I'm the Sales and Marketing Director for Cobra. A bit like Arvind, I've been working on the COVID-19 vaccine since March, actually, so slightly less time. But again, it's been transformative and just full-on hard work, really. It's been quite amazing.
Yeah, fantastic. Well, welcome to Philip. And Jack, going along my line of Jack.
Hi
welcome
there.
To Jack, a new entrepreneur on the team.
I am the newbie. Yes, thank you for having me. I'm Jack. I'm with a company called Ochre BIO. We started last year in the midst of the COVID pandemic, and it was when we were building the business. Not related to COVID, actually. Metabolic and liver diseases is our focus. And we're an siRNA therapeutics company, so could call that gene therapy. There's a bit of debate out there, but we'll, for the purpose of this, we'll consider ourselves that. Nice to meet you all. I'm looking forward to the discussion.
Fantastic. Moving along, Paul. I think I've got Paul on my left. Paul Hastings.
Yes, thank you. Yes, I'm Paul Hastings, and I'm President and CEO of Nkarta Therapeutics. We're a natural killer cell therapy company. And actually, we did a whole bunch of work last year to determine whether natural killer cells had a place in COVID, and we decided not to develop them there because they really don't have much of a place there. But we spent a lot of time making sure that we were right. We're working on natural killer cells in hematologic as well as solid tumors. I'm also the vice chair of the Biotechnology Innovation Organization known as BIO, where I spent a lot of time with cell and gene therapy companies. I'm also on the board of the Alliance, not on the board, I'm a member. One of my colleagues in the company is on the board of the Alliance for Regenerative Medicine, which is basically the cell and gene therapy trade association as well.
Fantastic. Thank you, Paul. And from a clinical perspective, we have my friend, or our friend, Panteli. Panteli, maybe you could introduce yourself.
Yeah, Tony, thanks for the opportunity, and great to see a number of familiar faces, on the screen today. So Panteli Theocharous, I'm the cell and gene therapy strategy lead for PPD, so global CRO. And it's been fascinating to see what's sort of happened in 2020 with regards to innovation. By background, I'm a bone marrow stem cell transplanter, so very early days of immunotherapy as we know it today. And certainly, Paul, to your point, had a real interest in NK cells. So great to see the field moving forward in that regard. So looking forward to the discussion today.
Fantastic. So let's start.
Right, there's Alan as well. Alan.
Where's Alan?
If you take yourself off mute.
I can't see.
Hello, I'm here. Good afternoon.
Yeah, sorry, Alan.
Very pleased to be joining this distinguished group. It's very, very exciting, always this conversation. So a few words of introduction. I'm a microbiologist by training, molecular biologist. I've also business development experience. I worked in BDNL for Eli Lilly, Pfizer, and Hoffmann La Roche. I founded a consulting company, NxR Biotechnologies, 10 years ago, and we focus on oncology, inflammation, rare disease, and neurology, but also on advanced therapies. And I've published 2 books on the first and, of course, the next generation, which shows a lot on engineered engineered CAR-Ts and NK cells, which was published by Elsevier in April last year. And we're working on a couple of more programs on MSCs, and another one focused on just on CAR-Ts.
Fantastic. Well, I'm going to kick us off with maybe Arvind giving us some, this to catalyze our discussion. I mean, the world has been talking about cell and gene therapy, and it's been trundling along. It's been incremental innovation, and then COVID came, boom. So just tell us a little bit about the story that you see, Arvind, for BioNtech.
So BioNtech, really, I mean, I joined the company even though the pipeline was predominantly oncology for infectious disease, right? It's primarily because it was a personal view that global warming was coming and there will be more infectious disease. But the time I joined, or still thought about joining, the company was 500 people, okay? And we were about, this beginning of this year, we were meant to start 12 clinical trials in oncology for different kinds, FixVac and personalized medicine in RNA therapeutics for customized cancer vaccines. And COVID came along and everything stopped. So all our partnerships have been very much with a number of big pharma. You can see them. We have done lots of different alliances, Genentech, Genmab, blah, blah, blah. And all of them are going, well, your deal terms are all based on the fact you're a first-in-class, untried therapeutic. And all of a sudden, in 12 months' time, the landscape's completely changed, and it will change the face of the pipeline and everybody else's pipeline because of the data, real-time data you're getting from the COVID vaccine. So it's been transformative, not just because it's a vaccine, but it's changed the landscape for accepting these kinds of therapeutics. And I think that's what's going to be really interesting in the years to come, actually. I think it's great that we've got the vaccine.
Maybe you could say, Arvind, I mean, what do you mean it's changing the portfolio? Because you mean that people are going to getting that almost these molecular approaches are quick, and that is that what's going to change it, or is it that people's swing from small molecules will completely move on to cell and gene? What do you
No,
mean?
I don't think you could move from small molecules. I think that you can customize drugs quite quickly. So because we have personalized vaccines to antigens that are specific. So if you know the RNA is safe and you can show that the target is specific to a cancer, then the approval process will be quite rapid, right? And so you will be able to produce, it won't work for everything, but when you're looking at unique antigens on a cancer cell, that these therapeutics will become more common. In the same way that CRISPR is everywhere now, that I think RNA therapeutics will find their place in disease.
Is this the gate? I mean, because I was CEO of Silence Therapeutics. I mean, when I was CEO, it was a bit like, well, actually, fashion was that it was quite flat and it was all about delivery. Do you think this has now uncapped the brilliance of our RNAi therapy?
I think it will make it so every day that people will be more willing to accept it. And I think the sustainability of the vaccine and how often you'll need it will in time prove the sustainability of this. If you have an antibody treatment every 6 to 8 weeks, but you have a vaccine, if you like, a cancer vaccine once a year, that's still cost effective, right?
So there's something happening. Now, can I bring in Jack then? Jack, just you've just heard that maybe the world's changing in your direction. I mean, what's your reaction to that?
Yeah, I think RNAi medicines are definitely on the rise. We as a company take a genomics-based approach to identifying novel targets, and I think that suits the RNAi space very well. As Arvind just mentioned, the programmable medicine is the direction we're heading, and by using genomics, you can very quickly identify what you want to focus on and identify stratification, so different patient populations, etc. So I think we're pretty thrilled. I'm supposed to say that though, but yeah, it definitely feels the field is turning towards siRNA once again from where you were back, Tony, with Silence not so long ago.
And Silence is doing well, right? So
yeah, Silence.
Yeah, I think
it's doing really well.
Yeah,
but it was interesting because when I was there, it was like living the 5-year fashion from before, and I think that but I'm welcome it. Paul, what's your thoughts from your eyes in the US? I mean, looking over at BioNtech and your own sort of interest with Moderna, and what's your thoughts on this?
Right. So I tend to not take a US-centric approach until maybe a few days from now when things change here a little bit. But I will tell you that we're very excited about BioNtech. And one of my best friends is John Maraganore, and the Moderna folks have done a fabulous job with flagship pioneering over here. I actually, yesterday, Alex Azar, our outgoing Secretary of Health and Human Services, announced they were going to open up the vaccines to a broader population because the distribution program that we set up here in the US has been miserably failing. And my husband and I signed up to get a vaccine, and I'm over 60, I have Crohn's disease. I'm kind of fitting the criteria, but we signed up for the vaccine, and I got an email from the Department of Health this morning saying I don't fit the criteria. So we still have some hiccups in our distribution policies, which, as you all realize, if we can make these things, if we can discover them and develop them, and then we can't get them to patients in a timely fashion, we're really not doing our job. So I think the main issue for these vaccines, there's no question, if you ask any citizen of the US that understands a little bit of science, they will say they would prefer the Moderna and the BioNtech vaccine to the other vaccines that are coming out. So I think there's a great understanding now that these are, state of the art. And I don't think there's any question that RNAi is the way to go. And you can just look at the success of Moderna and also Alnylam and the many companies that are in the space. So I think it's an exciting time for RNAi here in the US and globally in everywhere. And I think it's a terrific advancement.
Fantastic. And bridging the patients to my friend Panteli. I mean, what's your reaction from looking through it with clinical eyes?
Yeah, I mean, I think as the members of the panel have, articulated, eloquently, I think it's a focus on the science and the innovation. I think from my vantage point, being part of a global CRO, we partnered with many companies on a number of different clinical trials in COVID-19. And that I think really, that really was a motivation, how to work collaboratively, how to work collectively, and how to really focus on efficiency. I think, Paul, to your point, how we take the science, how we translate it, and how we bring that to patients. So, it requires a mammoth effort, but I think it's a highly collaborative effort. And I think that was really what was inspiring, just everyone coming together and working in partnership and collaboration. But yeah, it's the innovation that drives the interest of many different external stakeholders. But the patient, of course, needs to continue to be at the epicenter of, everything that we do.
Yeah, no, that's fantastic. And Alain from Switzerland, I think you're in Switzerland, no, France, with Alain, what's your thoughts?
My first thought is that just like the AIDS epidemics, the COVID-19 epidemic started a revolution in science. With AIDS, we started to learn more about retroviruses, about the link between oncology and immunology, and it eventually ended up in CAR-Ts, with emerging of various types of technology platforms like gene therapy. The advantage of, and there is always a positive development in any negative situation, and the positive development is definitely the emphasis it put on a new technology like mRNA for therapeutics. When I was at Roche, I was leading the large alliance between Alnylam and Hoffmann La Roche on preclinical. It was very, very complex to bring on board the various therapy area heads to use a new technology. There was a lot of internal resistance. To make a long story short, Roche eventually divested this, but 10 years later, the PCSK9 inhibitor that we pushed was bought by Novartis for $10 billion. So I think if we really look at this perspective, the big revolution that we are witnessing now is very much about the coming of age of mRNA therapeutics. And the biggest advantage that the whole world is watching, so it's a tremendous accelerator for this technology and also for related technology. So I think we have something to celebrate. Having said that, it's still a long journey before we get all the benefits from the technology. Now back to advanced therapies. The COVID-19 situation has also developed triggered a lot of interest for other platforms like MSCs to treat cytokine release syndrome. And I think all of this will be tremendously important in a couple of years, actually. What I'm trying to say is that there are reasons to be hopeful, even though the situation is obviously not what we wished for, but there are tremendous developments in science, and what we need to do collectively is to make the best advantage of this situation, which is unique, at least in our lifetimes.
Excellent. Now, asking Philip from a point of view, from your eyes, through your lens, what do you see with the BioNtech and the COVID vaccine world?
I guess as a CDMO on the manufacturing front, we see these as kind of platform technologies that essentially have been accelerated from what would have been quite a long-term, probably 10, 15, 20-year development into something that's been achieved in 11 months, which is amazing. So you've got these platform technologies, mRNA, DNA, viral vectors, that have all been targeting oncology areas mainly, and now they've been thrusted into the limelight and have certainly come to the interest of investors as new technologies for vaccine. And it's interesting that certainly investors are having to change their attitudes towards vaccines, because up till now, vaccines have been seen very much as the poor man's medicine. You know, we make them globally, and we make them cheaply. The returns aren't very large, and now you're seeing that actually these technologies can enhance the whole vaccine area. But also, I think the silver lining, we're trying to see a silver lining within this pandemic, is the fact that it has enabled the acceleration of these amazing new technologies.
Yeah, excellent. Going back to Arvind, I mean, if you look at the challenges that your group has dealt with, I mean, has it been what dose to give? Is it what the interval of doses in? You know, what is it getting enough active cases to treat? I mean, what were the real big challenges to you?
I have to say yes to all of them, but at different times and phases. What's nice to see is that, if you like, Moderna, CureVac, ourselves, there were many RNA companies doing the same thing and have all encountered the same thing. What is nice is that Moderna and BioNtech independently got the same data at the end in terms of efficacy. And the issues that came up are the manufacture of the lipids, the stability, the temperature storage, the cold chain, all those pieces. And we've each approached them slightly differently and ended up in the same place, ultimately. So we have the problem of the cold chain storage, of having it at -80, but our volume of injection is smaller. So I guess our focus, we took 4 candidates into phase 1, and we had a whole matrix of different buffers and a whole different set of storage conditions. So our focus on our, if you like, RTPP was low volume, right, low amounts of RNA. And Moderna's was high amounts of DNA, but a higher temperature storage. So it was a bit of give and take, but in the time we had to make different decisions all the time. And the decisions we have now are different, because now everybody wants vaccine yesterday. And we work with Pfizer very closely and trying to get all the component pieces, and this is why Philip doesn't have a life either, is having to get the manufacturing, the fill finish, all the components, the freezers. I've never bought as many -80 freezers. I didn't know that many you could actually purchase, right? So you sit there and you go, oh, I'll have 100 of those. It's a bizarre situation you're in, where you're taking and having to, the old day saying where you're driving the train while laying the track. I think this has been the issue all the way through. So unlike a lot of companies, I mean, we gambled all the way through. If it fell, it would have totally killed the company.
And
right?
I mean, as far as dose goes, I mean, you know, as a classical pharmacologist, like I know my friend Tom's on here, you know, we always think about kinetics and how you extrapolate the dose. I mean, how did you work out what dose to give?
So I think you look at the standard, the vaccine doses. Is it 3 weeks? Is it 4 weeks? Is it 2? We did all, I think we did a matrix of those as well. And I think so the 3 week was best for us, right? So we had to, and I guess because you're looking at an RNA, you're looking at expression of protein, that's your dosing. You don't need to actually have a viral assay. I mean, when we're working at Roche, you know, we're working on HCV, we're working on papillomavirus, you needed actually disease models. With the RNA situation, you're assuming the immune system will do what it's supposed to do. So now, are you making enough of the spike protein to get an antigenic response? Are you making antibodies in the mice? And then you move on. So your cycle time for secondary endpoints was, you know, okay. So it allowed that speed, I think.
So, I mean, just moving to Jack, I mean, hearing what Arvind say, I mean, are you thinking that a therapeutic or a vaccine which has got RNAi in it is always going to work? It's a slam dunk.
I didn't say that. Nothing's a slam dunk.
This is my question about, you know, this idea that you take the risk. The risk moves away from being, if you like, a discovery risk to the development risk, and you just shift your mindset. So you say, what I'm going to do is I'm going to put in 20 million, I'm going to take the risk. But it's a different mindset. And just Jack, your thoughts on that?
Yeah, I definitely think you're given the recent successes, like we've seen Alnylam get 4 in a row now, I think just this past week in the RNAi field, the approvals are there, and the modality, you have a lot more control or a lot more precision around the side effects. You have a better sense of the actual overall product profile. So I think you can take more calculated risks, but ultimately it is biology we're dealing with. You never know until you know. So I don't think that risk goes away by any stretch. But
yeah, moving on, Paul Hastings.
Yes.
Any thoughts from you about your reactions to those challenges and what Arvind was saying?
Yeah, I mean, I'd like to offer another slight challenge there, and that's, you know, the clinical trials, which, by the way, were extremely well done. They were diverse, they were balanced, they reflected the population that's going to be taking these vaccines. I was surprised to see, well, maybe not quite so surprised, but wondering how we're going to follow up, that all these vaccine trials were basically symptomatic endpoints, right? So if you took the vaccine, you have a 95% chance of having no symptoms. But we're now in an educational process where we're trying to explain to people that, you know, there's no clinically relevant endpoints that prove that you're going to be virus free. So we have to keep with the distancing and the masking and the essential worker restrictions we have around the globe. So I'm just wondering, I assume it was done for the sake of speed. And now, you know, what's the follow-up there? I think that's going to be a challenge because, as we all know, not everybody that goes to take a vaccine has the kind of knowledge that we do, or our families have, even by being associated with us. And so it's something we need to follow up on. And it's not to be critical because, again, you know, Alex Berla was on the board of BIO. He's a friend of BIO, and we had a conversation with him just the other day about what a great job that Pfizer did with BioNtech to get this vaccine out there. But that is a challenge, and just one I would be curious as to how people feel about it.
Any reaction, Arvind?
I mean, that's absolutely the case. I mean, you have to think about it. So my parents are getting the vaccine and go, but they feel that, well, I can still give the virus. But I guess the reactions of normal vaccines, when you have the flu vaccine, if enough people have the vaccine, then even though you can spread it, the person is also resistant themselves. And those are the normal dynamics. This case, we don't know. And I'm sure there'll be lots of follow-up and studies to know whether you pass on less. So at the moment, there are too many issues of dealing with the immediate cases, dealing with the variants, So those are the priority, but all these other things have to be followed on. And I think we do have at least a year follow-up on patients that do get the vaccine. So we are doing a lot of work, and I'm sure Moderna are doing the same and CureVac are doing the same, because this is a first-in-class product. We still have to be sure of the long-term effects, right?
Yeah. Let me bridge to Panteli. Any thoughts from you?
I think that these are all key points, aren't they? When we talk about how do we modulate the the power of the immune response, it comes down to an immunological phenomenon. And I agree. I think certainly from my vantage point, we saw almost a change in trend where many years ago there was a real focus on cancer vaccines and leveraging and developing technology. We're now starting to position ourselves in order to address the pandemic and with some of the other vaccines that are now available. And certainly I see a lot more opportunity. I see companies looking at their own platform and being able to develop these highly innovative therapies. I think the long-term follow-up, of course, is going to be important. So how do we develop registries to continue to collect the necessary data to maybe inform second, potentially third generation vaccines? I think to Arvind's point as well. And we know that that is a requirement in cell and gene therapy. 15-year follow-up of patients that have had a gene-modified cell therapy. That's a necessity. So I think there will be frameworks that we're going to need to really integrate to better understand the patient journey and to better understand the mid to long-term follow-ups. And
just Panteli and bringing Alain in, I mean, is this, I'm interested in whether you're going to get as I say, the risk moves to development because you're going to just put more things in now. And if it goes to development, then my interest is really, that's going to bump the price up. I mean, Alain, what's your thoughts about, you might have very cheap ones and you might have very expensive ones. I mean, Alain's thoughts on this sort of thing.
I mean, on the pricing of vaccines for mRNAs, I think that just like any industrial product, it's expensive at the beginning and a lot less later. So we will have this type of phenomenon going on as the mRNA vaccines get manufactured at scale and distributed at scale. Plus also there is no other choice for the large for the healthcare system. The price needs to go down, and we will need the help of CDMOs to find the solutions for that. Regarding vaccines, I think yes, the first generation is based on one spike protein, definitely, and we will probably. And I would say this is a tremendous advantage of the mRNA molecule rather than a protein. It's much easier to modify the APIs than it would be to modify a protein. Actually, it's in a vaccine. So I think there are many ways the potential of the new technology will reverberate and will very deeply impact healthcare in the coming years, way beyond the SARS-CoV-2, definitely. But in terms of manufacturing, I remain confident as the price go down, like for any industrial product, economies of scale, economies of learning, this will have a major impact.
Yeah. Now moving on to Philip. Philip, you're the CDMO. You're the one who's going to get us the cheap stuff. I mean, or cheaper stuff? But it does. If you're are we going to put more things into development? That's my question. And if so, I mean, that means there's a tremendous amount of capacity that's needed.
There is a huge amount of capacity, but I think drawing on Alain's point, I mean, yeah, it is always within the remit of the CDMO to look at ways of manufacturing more efficiently. I think that the challenge that we always face is introducing new technologies to facilitate that. I think as much as we are pioneering in terms of the research side, when it comes to manufacturing, people and companies, biotech companies, want to see that other people, other biotechs have trod the same route, down the same technology, use the same technology for manufacturing. So it does sometimes come a bit hard to bring in that innovation. Also, I think on the clinical trials front, we've shown that, you know, we're talking normally 10 years. That's a huge expense, massive expense. And if we've proven that we can do this in a year, then we need to relook at that system totally. Because as much as a CDMO can reduce the costs, you're looking at 10 years of clinical trials. And there's only so much a CDMO can do to reduce those costs. The main costs are in those clinical trials.
So back to Arvind, if you look at you know, speed, we're hearing about speed. What is it? Was it that you did just threw everything at it, at the vaccine, or was it?
We didn't actually take shortcuts in terms of development. We just did everything at risk. So you did multiple phase 1s to select your target. Then you said, okay, I'm going to do a phase 2A with multiple targets at risk and still not selecting your lead. That's a huge expense to do a 2A with multiple different RNA sequences, basically compounds. You're taking multiple compounds and saying, okay, this is the best profile in terms of antibody production or protein expression, if you want, of COVID, not even waiting for the antibody production. And then saying, okay, only at phase 3 do we select the lead. And even then we're going to do 2. I mean, these are huge trials that come at a huge risk to determine whether we would get it or not. I mean, in
a way, you know, when you've got an existential crisis going, you take the risk, but I mean, people aren't going to want to take that risk in the future, are they?
No, but I think what you might be able to do is, if you know the safety of an RNA is there, and you have a sense, as Jack is talking about, understanding the biology and having some surrogates to demonstrate the biological safety in an animal model, then your phase 1, 2A can maybe go more closely together. And so you can shrink the time. So you don't do a phase 1AB because there is no need for 1A. It's usually just a phase 1, but your phase 1A is a 2A. You do a 1, 2A together for these, and then you have stricter safety requirements. I think also, to Philip's point, it's the nature of COVID and having to manufacture so much so fast. He probably has greater scope to experiment than he's ever had before, because everyone wants to try and get more and faster than before. So there is potential to speed up production. And if the lipid part is similar in each case, then you already can say there's a precedent for safety.
Which is
the RNA, right?
The argument for the vector for the AstraZeneca one as well, isn't it? The famous plug and play, it's ready to go. But let me bridge to Panteli. I mean, this idea of we've always tried to bring trials down, but I mean, is this meaning that there'll be a greater pressure on real world data to tell us really what's going on with the drug?
I think it's going to be key. We know that, real world evidence, particularly in the advanced therapies field, is going to be critical to generate. And I think the encouragement to do that much earlier on in the drug development paradigm, is also something that, that's being realized. So how we develop strategy around clinical trial execution, and what are those parallel tracks? And the parallel tracks is really engaging with the regulators and the payers much earlier on in the life cycle of a particular asset under the ATMP umbrella. So I think there will be continued focus. And going back to your point, Tony, about the sort of the clinical trial framework, I saw, a real close collaboration with a number of these sponsors with the FDA. Peter Marks from the FDA was, very vocal in, several presentations around how important it was to partner early and be very clear on objectives, and let's work together in order to ensure that we're meeting these important milestones for patients. So it goes back to the point earlier around that true collaborative, collective effort.
Yeah, I mean, Alain, it's one of the things that's come up here is that the focus on the biotech being the generator of the idea and the big pharma being the facilitator. I mean, is this another public view of that the world has all changed?
I think from, again, just from my perspective, you know, there's some evidence to that. You know, we know, of course, particularly if we think about cell and gene a little bit more generally, big ideas being created in, you know, the sort of the academic centers of excellence, and then how do we translate those great ideas from non-commercial to commercial entity? But you're right, Tony, we saw, again, that partnership between biotech and big pharma, you know, leveraging to a certain extent capability and infrastructure in order to get, you know, these important milestones achieved. So I think it was just a reflection of the scenario to some extent.
But I think it's gone beyond that.
Sorry,
I mean,
sorry.
It's I mean, Tony, it's these, to get where we've got to has meant a collaboration with not only big pharma, biotech, manufacturers, it's been raw materials suppliers. I mean, it's the whole chain that have had to talk to each other, because we're all so intricately linked, and the pressure has been so great to achieve something that normally takes 10 years into 11 months, that the only way we've been able to achieve that is by sitting down with each other, throwing away the rule book of who knows what, who says what, and be very open with each other, saying, you know, these are the timescales that we need things. When can you do this? When can you supply that? And be very open and find solutions together to overcome them. I mean, because as we're all aware, you know, working on this, it has been a true collaboration right across the ecosystem, and wouldn't have been achieved any other way. And I don't think we've ever seen, or if you like, being forced into this opportunity to develop relationships along the whole length of that supply chain and development chain.
I have to support that we couldn't have done it without everybody. And everybody we talked to, manufacturing, glass vials, anything and everything, right? Brand name, the name for the, you know, it was offered for people, just came out of the woodwork to ensure that we help, that they wanted to help. And it's been phenomenal, actually, really.
And I think I'd just add to one comment to Arvind's point there. I don't think there was a compromise in any way, shape or form when it came to quality and patient safety. And I agree with the comments. I think it had to be a collective effort in order to get us to where we are today. Which
is brilliant. Now it's 45. I'm gonna because this is got a meaty and I know people will have questions. I think what we should do is open the floor up to others to join the group in the middle. Graham, maybe have you got any already in the chat, or shall I?
Yeah, we're gonna start off with Tom's got a question, followed by Gordon, and then Thorsten.
Yeah, Tom.
Yeah, you know, congratulations, you know, to all, you know, involved in, you know, this revolutionary technology, you know, whether it's RNA vaccine, the Oxford vaccine, etc. My question relates to the whole RNA area, but with the messenger RNA, is it pleiotropic in the sense that it's active against these different mutants? Because we know with the microRNAs, you know, that they control one microRNA can control as many as 200 different genes. So is this the case with the message, or perhaps the siRNA?
Who'd like to take that one? I think we could. Where's Jack? Jack, is Jack there?
Oh, he had to shoot at 45 past.
Yeah,
so
Jack, you've lost him. Maybe, I don't know, Arvind or one of the others would like to take that.
I mean, it's an mRNA, so the RNA sequence to the coat protein does work with different variations of the COVID. So it may not be true in all cases. In this case, it is, and I don't know whether the Moderna one probably equally has some cross-reactivity, but we don't know, and that will be the case over time. The more specific you make it, maybe therefore it will have less cross-reactivity. And it also depends on the length of the RNA you're putting in as well, right?
Yeah.
Do you have any comments, Paul, on that at all? No? Yeah.
In terms of manufacturing, no. I mean, I wanted to move to manufacturing. I actually wanted to move out of COVID for a minute, but I'll wait my turn for that. I
mean, we're delighted to move out of COVID, but just while we've got a couple hot questions I can see coming up in the screen. Graham, do you want to take the next one?
Yeah, Gordon, if you'd like to ask, you've got a couple of quick ones, haven't you?
Yeah, I'd just like to echo Tom's congratulations. But how did BioNtech actually start working with Pfizer? You know, getting the lawyers signed. How did you find one another? Is there a story?
Okay, so that's part of the reason we're with Pfizer is we already had a collaboration with them for flu. So we didn't have to sell the technology. We'd done all that bit. So we already had a relationship with them. So we didn't start directly from scratch. And actually they approached us. So it was sort of, we'd started working on it and we thought, okay, well, we can't do this on our own. And then they came at the same time, sort of thing. So it's because they knew, and they'd spoken to Ugur, who's a very strong scientist. So they knew him, they knew the technology, they knew the team, and we already had quite a good collaboration ongoing in the first place. So it was a slightly, we didn't start from scratch with Pfizer.
Great. Okay, we're going to move on to Thorsten, please.
Hi. Look, my question is very much around the clinical trials you mentioned earlier. So you were saying that you carried them out on risk, on high risk strategy. So do you think that is something we're going to see more often in the future, that clinical trials being run on a more risk, on a higher risk stake model than?
Yes, I'd say Corona is a bit of an exception. But I think there is justification potentially with a VC strategy if they say, okay, the biology is very well established. If you did another rheumatoid arthritis vaccine, that potentially the animal models are really clear. So if the biology is very straightforward, then you can say, okay, therefore it's probably worth a risk because of partnering, licensing. The old adage when I was at AstraZeneca, they would say every day you can get something faster to the market, the amount of revenue you're making, because your patent life is extended, right? So there's a lot to be said for earlier to the market, but it's a very much a risk managed situation.
Remember, Arvind, that mantra created actually a lot of drugs that started to fail. It's because, so I think the mantra, the interesting mantra actually is, a bit like we want to know that this drug works and then we'll just put it into development. But let me ask, I want to ask Paul, because just to make a more general comment about other, because I can sense that you've got something else to say. So say it.
Yeah, well, I wanted to talk about manufacturing, but to that point, one of the things I want to remind everybody, there are roughly 600 programs in development right now for COVID. And so one of the issues that's going to come up here is background therapy and doing better than what's out there today. It's going to be tough to beat a 95% response rate. Now, when you're looking at therapeutics and combinations for treatment, I think that's a different story, but I think there's going to be some challenges there, but I think we'll certainly overcome them. You know, I just really wanted to talk about, there was this discussion about collaboration, which, by the way, I absolutely celebrate the collaborative approach that everybody used. And I think, you know, BioNtech and Pfizer were just spectacular in that collaboration. It's not easy working with big pharma. We all came from big pharma, so we know this. It's not because they're bad people. It's just, it's a bigger organization and we'd like to move fast, and you guys did it. And, you know, those folks at Pfizer were very determined they were going to do it too. But, you know, one of the things we made a decision of very early on, we had $3 million in the bank when we went into our Series B crossover, and then our $300 million IPO shortly after that. But one of the early decisions we made was, this is going to take a lot of money. So let's raise it. Let's raise it with the right investors. And let's not rely on CDMOs for manufacturing. Not because they're not good manufacturers, but because the whole process development, CMC, you know, supply chain issues, if you can control them yourself, great. And particularly if you have a cost of manufacturing, that we do, we have a $2,000 cost of manufacturing per dose. Now this is for a NK cell for cancer, so that's a pretty decent cost of manufacturing. So we built a 3,000 square foot clinical GMP facility and are currently planning out a 30,000 square foot commercial GMP facility. And companies are doing these things earlier in order to maintain their trade secrets, in order to control manufacturing. And what's fascinating about the space in cell and gene therapy is you get value for that, even at the early stages today, whereas you didn't before. So I think the world is waking up to what we do takes upfront investment, and that upfront investment, if you have people sort of begging, for incremental amounts of cash along the way, it's going to be harder to do that. But if you can do that, I think that's the way to go on manufacturing, and still rely on, the folks in the middle for the raw materials and things like that, but have more control over manufacturing. And I think that you're going to see many, many more companies globally doing that going forward.
Yeah,
I think that's a challenge in Europe, because you don't get the big money, right? Investors, I mean, I don't know about anybody else here in your UK or Europe, but VCs here don't give those large chunks of change that you can get in the US. You just don't get it.
Yeah, no, I lived there as well for 8 years. I understand where you're coming from, but I also think that the recognition that innovation doesn't, I mean, people talk about how, we're the kings and queens of innovation in the US. That's horse manure, as you all know, right? So I think there's a recognition by what I'm seeing in now global organizations working on the venture side that, you can go and find that innovation in Canada, in Europe, other parts of the world, and invest in it, as long as there are regulations that allow you to do so. And there are many countries where it's more difficult to get, outside capital. But I think I agree with you. It depends on where you are geographically. Even in the US, depends on what cluster you belong to and where you are, whether people will invest in the company or not. So I totally relate to that and totally agree with that as well.
Yeah, let's move on to that. That's great. Thanks, Paul. Let's move. I've got Emma. Emma.
Yes.
Question.
Emma Banks. Yeah.
Where's Emma?
Hi. Thanks so much.
Here she is. Once she arrives, you can't miss her, can you? Hi, Emma. Nice to see you. Love the hair. Love the hair.
Really great session. Thank you so much. It's been fabulous. I just had one question and well, two questions actually. First of all, just in the mainstream media, we're obviously hearing a lot in the UK, particularly at the moment, that we're ready to vaccinate, but there's just not enough supply. So I was just interested in what further work's perhaps being done to look at improving vaccine yield or output, if anything can be done. And also just interested in any comments on the COVAX initiative, because clearly, unless we vaccinate everybody, we're not actually going to get out of this. So just those two would be great, and probably not that straightforward. But
one comment is, it's been a very hard line to not be political, right?
Yeah, sure.
Our goal is to get a drug to market. It is to governments to decide how to get it to people. It is up to people to decide they need to be vaccinated. So that means we have to be there. With respect to getting more vaccine out, I think Philip can agree with me here. There's a shortage of everything because everybody wants vials, wants syringes. Mm-hmm. So the easiest thing we've done, we've just got approval in Europe. I don't know if that includes UK anymore, to do the six doses per vial. So that means we get 20% more doses per vial, right? And we're doing everything to try and increase yield, increase, the amounts, double the batches. So, but the problem is also getting the supply to match it. Mm-hmm. It's the difference here, I think, that people is when you do with a small molecule or an antibody or even a protein, it's one thing. Mm-hmm. There are so many components to this that really it's a challenge to get it all together in the same place, to stick it in a vial. Yeah. So that is the challenge. But guarantee that not only us, everybody is working together to try and come up with ideas, and everybody is still offering, taking things off, but other big pharma are offering to say, we'll stop this line so you can fill and finish, which is amazing. And they've never been CROs. So we have to realize that all the pharma industry, biotech and pharma together, have really come together and are offering and taking and doing things well beyond things they've ever done before to support this. Yeah. You'll see changes.
Yeah, and just to just to add to Arvind, I would say it also emphasises the importance of having the opportunity with multiple vaccines, because then that spreads the pressure in terms of manufacturing, because they do use slightly different resources, if you like, for that manufacture. So, because I know the media are quite keen to put one vaccine against another, which is the best, which is, at the moment, in terms of supply, we need as many vaccines that are made in different ways in order to ensure that that supply chain continues, because there is such stress on the supply chains. So the more vaccines that come online and use different manufacturing techniques, so much the better.
Yeah. I'm mindful of time, Graham, but
yeah.
I'd like to really ask each one of our colleagues just to say a couple of final words to the group. I mean, Arvind, thanks to, if you can send all of our big thanks from the Agile leaders and the life science community that we represent. Thank you to all your gang for doing all the great things. But any last words from you, and then I just go around the group, any last words?
I'd say that it's been great to be part of this at BioNtech, but actually everybody just did their job. And I think we, at the beginning of this year, thanked everybody else, because we couldn't have got here without everyone else. So, and there's a certain amount of luck. I mean, whatever you say in this world, we were lucky as well. It could have equally been Moderna, it could have equally been AstraZeneca first. It's just, we've been lucky to be the first. And I think the thing I'm most proud of is the first vial that went to a UK person is going in the Science Museum. And as London is my home, I'm very excited to be able to one day be able to go home and go to the Science Museum to see that first COVID vial as on the exhibition. And I think it's a piece of history. I just hope we all get to go out soon. That's my last statement.
Fantastic. Let's move just to go to, where's Jack? Is Jack back with us?
No, he's not. No, he's gone away.
So let's move to Paul. Last thoughts from you, Paul.
Yeah, I would like to thank Arvind and the BioNtech team for having recaptured the reputation of our entire industry this year. I think that's the single most large contribution I think this industry has made in a very long period of time, and has done this collaborative approach has done a huge favor for all of us.
Fantastic.
Yeah, absolutely.
Panteli.
Yeah, just to reiterate, I mean, I've been inspired by, the focus on the science, and how we can be innovative. But as we've said many times, it's also about how we come together collectively and collaboratively, and we bring this to, to patients. So just that spirit and enthusiasm and motivation of how to work together and bring down barriers, for me, that was all inspiring, and hopefully, we continue to, showcase that character and that personality moving forward.
Brilliant. Alain.
Well, I just have a few perspectives. I think what this tells me is that the process that we have in the pharma industry has been greatly accelerated. I would say it's the same thing, but at a much, much higher speed. And it's quite fantastic what a little bit of sense of urgency can achieve. Another thing I think to consider is the learnings that the industry needs to make after this crisis is over, because we can probably reinvent how we develop novel drugs. Taking risk early in clinical trials, we can think of basket trials, we can think of a lot of different types of doing this type of studies. But I just, as we were discussing about the risk taken to validate various mRNA vaccines, I could not stop thinking about pembrolizumab. And Merck did about, if I recall, 25 different phase 2 trials with pembro to find the right cancer, the right indications. And I think this is perhaps part of what we need to consider to get those quick wins. We may need to take more risk in terms of clinical trials and perhaps rethink the overall approach.
Which means more capital, which we know. Now moving on to our next, to Philip.
Yeah, well, I think it's both the collaboration side. I know Big Pharma, I know I've spoken to them, they do risk assessments, collaboration, on risk assessments in terms of pandemics coming through, and I don't think any of them imagined the depth or the speed of the collaborations that they saw happening. And then the second one for me is that there is a silver lining in this. We've seen these new technologies being accelerated beyond anyone's belief, and that has to be ultimately beneficial to patients.
Excellent. Thanks very much.
Well, I'm mindful of time. So,
yeah, we're going to go. We're going to have a break until 10 past. So, you know, if everybody disappears.
I just want to thank everyone. First of all, I want to thank our panel. They're more than a panel. Thank you very much.
Yeah, thank you very much.
We've enjoyed it very much. Graham, over to you.
Yeah, absolutely. So we're going to have a 7-minute break. We've got another panel coming up just talking about business development and the challenges and how to overcome the challenges during these times of COVID. So we've got a great panel there. And then we're going to do 30 minutes of breakout so you can meet the people in the room. So please stick around. Very important. Arvind did a great job today. She is back on Friday as well. So if some of those questions you didn't get to ask today, I'm sure we can do that on Friday. So yeah, enjoy your 5-6 minute break.
See you in 7. Fantastic.